Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from AstraZeneca.
This program is presented by Medscape Education Global.
Hello everyone, and welcome to this program entitled The Evolution of Antibody-Drug Conjugates for Gastric and G-Junction Cancers. We'll focus on the latest clinical data. My name is Erik van Cutsem, and I'm Professor of Medicine at the University of Leuven in Belgium. And in this activity, Dr. Koei Shitara from Japan and Dr. Jeffrey Koo from New York and myself will discuss the increasing role of ADCs in treating gastric and G-junction adenocarcinomas. We will focus in particular on those targeting HER2 and Claudin 18.2, including the latest data from ASCO 2025. In this chapter, I'm going to give an introduction to antibody-drug conjugates for gastric and G-junction cancers. First, to start off, if you look at the guidelines and the treatment algorithm recommendations, and this is the recent ESMO recommendation, HER2 positive and HER2 negative cancers have a different treatment algorithm. In HER2 positive gastric and G-junction adenocarcinomas, it's standard to start with the combination of a chemotherapy, a cytotoxic doublet, in combination with trastuzumab, and in patients with a PDL positive tumor to add a checkpoint inhibitor. Pembrolizumab is in the guidelines. In patients with a PDL negative tumor, it's a chemo doublet plus trastuzumab is the standard in first line. If you look and focus on the HER2 negative cancers, there we look at different biomarkers, MSI high or MSS tumors, PDL positive or PDL negative tumors, Claudin positive or Claudin negative tumors, and these different biomarkers determine the treatment choice for what we add to a chemotherapy. In second line, treatment depends, of course, what we did in first line treatment, but in second line treatment for the HER2 positive tumors, an antibody-drug conjugate, the only one in gastric cancer that is approved, is being recommended. The treatment algorithms, trastuzumab deruxtecan, MSI high tumors. If a checkpoint inhibitor was not yet used in first line, pembrolizumab is used. And in the other tumors, can be a combination of paclitaxel plus ramucirumab or ramucirumab alone, depending on the general condition of the patients. And in third line, trifluridine tipiracil is approved in third line treatment. And for the HER2 positive patients, if trastuzumab deruxtecan was not yet proposed as a treatment option for patients in second line, it is approved in patients in good condition with HER2 positive gastric adenocarcinoma. So major advances have been made, having an impact on the outcome for patients, but this clearly shows also that there is a high unmet need for new treatment options for patients. And one of the promising classes of agents are the antibody-drug conjugates. An antibody-drug conjugate is typically composed of a monoclonal antibody, a cytotoxic payload, and a chemical linker. And in an ideal situation, an ADC drug remains stable in blood circulation, reaches the therapeutic target accurately, and eventually releases the cytotoxic payloads in the vicinity of the target. And the target antigen expressed on tumor cells is the navigation detection for ADC drugs to identify tumor cells, and it also determines the mechanisms for the delivery of cytotoxic payloads to the cancer cells. Focusing on the mechanism of action, and you will hear more clinical data later on, what is important is the delivering of the cytotoxic payloads engage with immune effector cells to elicit anti-tumor effects through antibody-dependent cell-mediated cytotoxicity, ADCC, antibody-dependent phagocytosis, and complement-dependent cytotoxic activity. The antibody component of ADCs can also interfere with target function and dampen downstream signaling to inhibit tumor growth. We see different ADCs targeting Claudin 18.2, HER2, c-Met, TROP2, and some other targets also are under investigation in gastric cancer. And we hope, of course, that several of these will be active and effective for patients with gastric cancer expressing some of these different antigens. Trastuzumab deruxtecan is approved in gastric cancer, and also it is now by the FDA a tumor agnostic approval. By EMA, it's restricted to HER2 positive gastric cancer and also to breast cancer. In gastric cancer, it's approved by EMA for HER2 positive gastric cancer in second line and beyond, based on the data of the gastric Destiny Gastric 01 and 02 study. Disitamab vedotin is another HER2 targeted ADC that is mainly studied at the moment in China, with also some interesting activity. And then also the field of TROP2 is really a new expanding field also in gastric cancer. Sacituzumab tirumetecan and datopotamab deruxtecan are two ADCs targeting TROP2. Several other ADCs targeting Claudin 18.2 are in development. Different clinical studies are running with, for instance, ACD 0901 or CMG 901. You will hear more details about that later on in this webinar, and also some others such as LM302 are under development. And then also interesting in gastric cancer are the c-Met targeting ADCs. ABBV 400 is one of them that is under development, where a robust clinical trial program is running. So as you can see, interesting, promising data with the new mechanism of action of drugs, new classes of agents, antibodies with the linker, with different cytotoxics. So thank you for your attention. Please continue on to listen to the next chapter entitled HER2-directed antibody-drug conjugates, where you will hear more clinical data. Thank you very much.
Hello, I'm Kohei Shitara, Director, Department of Gastrointestinal Oncology, National Cancer Center Hospital East in Japan. In this chapter, I'm going to discuss HER2-directed antibody-drug conjugate, including the latest clinical trial data. So let's firstly briefly review the current management landscape. The ESMO guideline recommend first-line therapy in HER2-positive disease, a combination of doublet chemo plus trastuzumab with a PD-1 inhibitor, especially pembrolizumab added for PD-L1 positive or CPS1 or higher patient. For second line, trastuzumab deruxtecan or TDXT is now recommended for patients who have received prior trastuzumab-based regimen. Similarly, in the third line setting, TDXT is recommended for patients who didn't receive this regimen before second line. Unlike ESMO, the ESMO Asia or PAG guideline recommends TDXT as a third line therapy for patients who have received prior trastuzumab-based therapy. So these guidelines reflect the advance of treatment landscape with ADC having an increasing importance role. So TDXT is an anti-HER2 ADC. It consists of humanized anti-HER2 IgG1 monoclonal antibody coupled to a topoisomerase 1 inhibitor payload by a cleavable tetrapeptide-based linker. TDXT has several key characteristics: a higher drug-to-antibody ratio to approximately 8, higher than most other ADC. Also, the payload is membrane permeable with a short systemic half-life time. This payload can be released from a HER2 positive cell and induce a bystander anti-tumor effect, allowing it to affect nearby tumor cells regardless of HER2 expression. TDXT is now approved by FDA, EMA, and PMDA based on the Destiny Gastric 01 and 02 studies conducted in Japan and Western patient population. Of note, there are no phase 3 study and no randomized study to compare TDXT and LAMPAC as another standard second line regardless of HER2 status. Therefore, Destiny Gastric 04 or DG04 was conducted. This was a global multi-center randomized phase 3 study comparing TXT 6.4 mg per kg every 3 weeks versus LAMPAC in patients with HER2 positive gastric or GEJ adenocarcinoma who had disease progression after trastuzumab containing therapy. Key eligibility criteria included HER2 positivity by central or local assessment using re-biopsied sample after disease progression of trastuzumab. The reason is to exclude patients with loss or reduced expression of HER2 after previous trastuzumab, which was one of the characteristics of gastric cancer. Randomization was stratified by HER2 status, geography, and time to progression on first-line treatment. The primary endpoint was overall survival, with secondary endpoints included progression-free survival, response rate, disease control rate, duration of response, and safety. The primary endpoint of overall survival was met with a median overall survival of 14.7 months with TDXT versus 11.4 months with RAMPAC, showing 3.3 months difference in median survival with a hazard ratio of 0.70 and a p-value of 0.0044, which was lower than predefined significant threshold. This difference represents a 30% reduction in the risk of death with TDXT. The survival curve separated earlier and maintained separation throughout the following follow-up period, indicating consistent benefit of TDXT. PFS was also statistically significantly improved by TDXT with a hazard ratio of 0.74 and a p-value of 0.0074. Median PFS was 6.7 months with TDXT versus 5.6 months with RAMPAC. The PFS curve also showed early separation and maintained until follow-up, indicating that the benefit of TDXT manifests quickly and persists throughout treatment. The confirmed objective response rate was significantly higher with TDXT at 44% versus 29% with RAMPAC, a difference of 15% with a significant p-value. Duration of response was also longer with TDXT. The median duration of response was 7.4 months with TDXT versus 5.3 months with a control RAMPAC. The probability of maintaining response at 6 months is 58% with TXT versus 36% with Rampac, showing more than 20% difference. The disease control rate was also higher with TXT, with 92% versus 76%. So looking at the safety profile, grade 3 or higher treatment-related adverse events occurred in 68% with TXT versus 74% with Rampac. Incidence of serious adverse events were almost similar. The rate of those modifications were almost similar. 12% patients with TXT and 13% with Rampac discontinued treatment due to treatment-related adverse events. The most common drug-related adverse events with TXT included fatigue, myelosuppression, and GI toxicities. In contrast, common toxicity with Rampac included myelosuppression, fatigue, neuropathy, and hypertension. The allergy or pneumonitis event in the TDXT were observed in 14%, but most of them were with grade 1 and grade 2, and only one grade 3 event. So there was no grade 4 and 5 event. In contrast, 3 patients experienced grade 3 or higher ILD with Rampac, including one grade 5 event. So overall, these results support TDXT as a preferred second-line standard of care for patients with HER2-positive gastric and GEJ cancer. So next, let me briefly introduce Destiny Gastric 03 study, which explored the first-line combination approach with TDXT. This phase 1-2 study evaluated various combinations with TDXT and chemotherapy with or without pembrolizumab to identify optimal regimen for further development. The recent ASCO GI update demonstrated that using a lower TDXT dose of 5.3 mg per kg combined with reduced 5-FU capecitabine and pembrolizumab maintained high efficacy with response rate of 73% with IC3 plus and 83% in IC2 plus and H positive. So these combinations were not without toxicities. The TDXT combination with 5-FU and capecitabine and pembrolizumab had a higher rate of grade 3 or higher adverse events. The report of ILD or pneumonitis were within the range of previous study of TDXT. However, reduced dose of TDXT and capecitabine or 5-FU showed manageable toxicity profile with this dose modification. So looking forward, Destiny Gastric 05 trial is now evaluating TDXT plus chemotherapy with or without pembrolizumab versus standard chemotherapy plus trastuzumab with or without pembrolizumab in the first line. This will help determine if the benefit seen in later line setting can be extended to the first line therapy. Of note, patients were stratified by CPS since current standard care is determined by CPS, and the DGO3 also showed enrichment of benefit of pembrolizumab in CPS 1 or higher population. So shifting gear focus to emerging therapy, this time of Belotine represent another promising HER2-directed ADC. While not yet globally approved, it has received approval in China and is being actively studied internationally. This Belotine differs from TDXT in several ways. It uses MMAE as its payload, which is a microtubule disrupting agent rather than topoisomerase 1 inhibitor payload. It has a drug-to-antibody ratio of approximately 4, lower than that of TDXT, but still sufficient for potent activity. Like TDXT, it employs a cleavable linker and demonstrates a bystander effect. It is currently approved in China for patients with HER2 overexpressing locally advanced metastatic gastric cancer who have received at least 2 prior systemic chemotherapies. In a phase 2 single-arm trial in Chinese patients, this combination of this vedotin with tislelizumab as a PD-1 inhibitor and S1 in the first-line setting showed very promising efficacy with a response rate of 89% and a median PFS of 13 months, with and 18-month survival rate was 68%. The rate of grade 3 or higher adverse events were 65%, with the most common adverse events being neutropenia, neuropathy, and anemia. Data was recently presented from a phase 2 part of a phase 2-3 randomized multi-cohort study of first-line listamab vedotin plus toripalimab as anti-PD-1 and chemotherapy or trastuzumab in patients with HER2-positive or HER2 median or low expressing gastric and GEJ cancer. And this study was also conducted in China. In the HER2 overexpressing cohort, the response rate was 82% for this vedotin plus toripalimab and trastuzumab, compared with 69% for toripalimab plus trastuzumab and CAPOX. And the median PFS was not reached with new combination compared with 14 months. In the HER2 median low expressing cohort, response rate was 72% for this vedotin plus toripalimab and CAPOX, compared with 48% for toripalimab plus CAPOX as a control arm. In the median PFS was 9.9 months versus 7.2 months. These efficacy results demonstrate the potential role of this combination approach in the first-line setting. Any grade AEs occurred in 100% of patients. Grade 3 or higher AE occurred in range of 73% to 100% of patients, depending on the cohort. The most common grade 3 or higher adverse events were diarrhea, myelosuppression, and hypokalemia. The safety profile was consistent with no effects of this agent and considered as manageable. So to conclude, what are implications of these latest data for our clinical practice? First, the current treatment sequence may need updating based on these data. These data strongly show the survival benefit of TXT in the Destiny Gastric 04 trial, establish its role as a preferred secondary option after progression of trastuzumab-based treatment. Second, we must consider how best to mitigate and manage the specific toxicity associated with this agent, particularly ILD or pneumonitis with TXT. To follow monitoring guidelines, early intervention strategy, and patient selection criteria will be crucial as these agents become more widely used in clinical practice. And third, we should ongoing consider whether these agents or ADC might be more effective in combination with other agents. This in Gastric 03 and the recent vedotin study suggested promising activity when combined with immunotherapy and chemotherapy, potentially offering a synergistic effect. The impressive result raised the question whether these agents should move to first-line therapy, either alone or in combination. The Destiny Gastric 05 trial will help answer this question, but the strong efficacy signals seen in later line certainly provide a rationale for evaluating earlier use. So in summary, HER2-directed ADC represent a significant advancement in the treatment of gastric and GEJ cancer with a robust evidence supporting their efficacy and manageable safety profile. Thank you for your attention. So please continue on to listen to the next chapter titled Claudin 18.2 Directed Antibody-Drug Conjugate.
Hello, I'm Jeffrey Koo, Associate Attending Physician and Head of the Esophageal Gastric Section, Gastrointestinal Oncology Service, and member of the Cellular Therapy Service in the Department of Medicine at Memorial Sloan Kettering Cancer Center in New York. In this chapter, I'm going to discuss Claudin 18.2 directed antibody-drug conjugates. Firstly, what is Claudin 18.2? It is a tight junction protein that's exclusively expressed in normal gastric mucosa cells, but is also retained in most esophageal gastric adenocarcinomas. In normal gastric mucosa, claudin 18.2 is typically buried within the tight junctions. However, during malignant transformation, loss of gastric mucosa cell polarity may result in claudin 18.2 becoming more exposed and thus accessible to therapeutic antibodies and other strategies. How is claudin 18.2 expression measured? Currently, this is with immunochemistry, and the definition of positivity at this time is IHC 2 to 3 plus, so moderate to strong staining in at least 75% of cells using the 4314A antibody. So what is currently approved for targeting this biomarker? The currently only approved drug and first-in-class drug is zolbetuximab. So zolbetuximab is a monoclonal antibody that binds to claudin 18.2 with high affinity, and it mediates antibody-dependent cellular cytotoxicity and complement-dependent cytotoxicity in claudin 18.2 positive cells. Zolbetuximab was approved by the EMA and FDA in 2024, and specifically it's approved in combination with fluoropyrimidine and platinum-containing regimen for the first-line treatment of patients with advanced HER2-negative esophageal gastric adenocarcinoma that is claudin 18.2 positive, using the antibody and the criteria that I just described. It is also included in the latest NCCN and ESMO guidelines. Despite the success of zolbetuximab, the question is, why are additional therapies needed to target claudin 18.2? Well, newer therapies may address resistance mechanisms, many of which we are only beginning to elucidate and understand with zolbetuximab. Newer therapies may have a different toxicity profile, and newer therapies may enable the targeting of lower expressions of claudin 18.2. Specifically, what is the rationale for targeting claudin 18.2 with antibody-drug conjugates, or ADCs? Well, the benefits of ADCs are that they have high targeting specificity. They are able to deliver a potent payload directly to the tumor cells, thereby minimizing toxicities in normal tissues. In fact, many of the chemotherapy payloads of ADCs are so toxic that they cannot be given systemically on their own. Finally, ADCs may also exploit or incorporate the bystander effect, and this is where the chemotherapy payload can diffuse into neighboring cells that could be claudin 18.2 negative. So there are actually multiple ADCs that are being looked at against claudin 18.2, but I'm going to focus on studies that have completed phase 1/2 results and indeed are actually entering phase 3 evaluation. And the three drugs are AZD0901, previously known as CMG901, IBI343, and LM302. So to start with, we'll talk about AZD0901. So this is a claudin 18.2 targeting ADC that has a humanized anti-claudin 18.2 antibody that's conjugated to the chemotherapy payload MMAE, which is a microtubule inhibitor, and this has a drug-to-antibody ratio of approximately 4 to 1. AZD0901 has been evaluated in a phase 1 trial of dose escalation and dose expansion. This was a study conducted in China and did include patients with esophageal gastric cancer. 27 patients were treated in dose escalation, and 107 patients were treated in dose expansion. So the findings are common to all ADCs, is that the most common grade 3/4 toxicities are primarily hematologic, related to the chemotherapy payload. So in this case, grade 3/4 neutropenia occurred in 20% of patients, anemia in about 15% of patients. In addition, 10% of patients had grade 3/4 vomiting, and this is both because of the chemotherapy, but specifically because anti-claudin therapies can also cause gastritis. In this study, there was one treatment-related death, and a dose of 2.2 milligrams per kilogram every 3 weeks was the proposed recommended phase 2 dose. The objective response rate was 29%. Subsequently, there is an ongoing study called the Clarity Pan-Tumor 01. This is a phase 2 study that has several components, and sub-study 1 is specifically evaluating AZD0901 monotherapy in patients with claudin 18.2 positive gastric cancer who have progressed on 2 or less prior lines of therapy. Primary endpoints are to assess objective response as well as better characterize the safety and tolerability of the drug. The study is currently ongoing, but in the meantime, a phase 3 study has launched, and this is a randomized, open-label but sponsor-blinded phase 3 study that's evaluating AZD0901 versus investigator's choice of therapy, after one or more prior therapy for patients with advanced esophageal gastric cancer. Patients actually are initially randomized to 3 arms, and that's 2 doses of AZD0901 and investigator's choice. And subsequently, once the optimal dose of AZD0901 has been identified, the study then becomes a 1-to-1 randomization with that dose of AZD0901 and investigator's choice. The study has 2 dual primary endpoints. The first is progression-free survival in the intention to treat population, as well as overall survival in the 3rd line and beyond patients. Now, notably, this study allows prior treatment with a naked Claudin 18.2 antibody, i.e. zolbetuximab, and it does require biopsy either within 24 months of study entry or a fresh biopsy to confirm Claudin positivity. The next ADC I'll talk about is IBI-343. So this is a fully humanized anti-Claudin 18.2 antibody, and what's unique about it is that there is silencing of the Fc component of the IgG1 molecule. In addition, it's attached to a topoisomerase inhibitor payload, exatecan. So again, this has been studied in a phase 1 dose escalation and expansion in China as well as Australia, and there were 89 patients who were eligible for efficacy assessment. The response rate, again, is quite similar across all of these drugs. The response rate was 32.6% with a disease control rate of about 81%. Again, most common toxicities were myelosuppression, which were seen in almost half of patients. In this case, GI toxicities, grade 3 GI toxicities, including nausea, vomiting, and anorexia, were reported in less than 2% of patients. Treatment-related toxicities led to treatment discontinuation in 2 patients, and none of these toxicities led to death. Based on the phase 1/2 results, there is a randomized phase 3 study that's recruiting in China and Japan, and it's evaluating IBI-343 versus, again, investigator's choice in patients who have received at least 2 prior lines of therapy. The study also has dual primary endpoints of progression-free and overall survival. Finally, the last ADC that I'll discuss today is LM302. Again, this is an ADC that's conjugated to MMAE, so conceptually somewhat similar to AZD0901. The phase 1 and 2 dose escalation and expansion was performed exclusively in China, and 97 patients were treated in the dose expansion phase. Once again, very similar toxicities. Grade 3 or more toxicities occurred in 45% of patients. Most common toxicities were neutropenia, about 20% of patients, as well as leukopenia, also about 20% of patients. Response rate was 30%, disease control rate was 75%. The median progression-free survival was a little bit more than 7 months. Median overall survival has not been reached in this cohort, but the 6-month overall survival was 95%, which is certainly very encouraging. There were updated data regarding the activity of LM302 plus toripalimab, which is an anti-PD-1 antibody, as first-line treatment for patients with esophageal gastric cancer. 43 patients were enrolled from Australia and China, and the primary endpoint was PFS. Of the 43 patients, 42 are efficacy evaluable, and very encouragingly, the results show a response rate of 64%. And in fact, in patients with Claudin 18.2 expression of 25% or more, and PD-L1 CPS of 1 or more, the response rate was nearly 80%. With a median follow-up of more than 9 months, median progression-free survival has not been reached. Similarly, median overall survival has not been reached, but the 12-month overall survival rate is 77%, all of which is very encouraging. So on the basis of these data and the initial data, there actually is an ongoing phase 3 study of LM302 that's recruiting in China only. Like the other studies, it's comparing LM302 versus investigator's choice in patients who have progressed on 2 or more prior lines of therapy. Once again, primary endpoints are both PFS and OS. So to summarize, there are several anti-Claudin 18.2 ADCs that are being evaluated in phase 3 studies, both globally as well as in China, in the 2nd line and beyond setting. So far, the data would suggest that they have similar efficacy and toxicity profiles, and certainly the results of these studies are eagerly awaited to see if we can build on the momentum from the approval of the first-in-class drug zolbetuximab in the first-line setting. Thank you very much for your attention, and thank you for participating in this activity. Please continue on to answer the questions that follow and complete the evaluation.
This program was presented by Medscape Education Global.